today as well. So today's topic, industry expert webinar, Skin Senses, the power of olfactory receptors. Now, this is really interesting. This is new science that's being revealed. And what we're going to be talking about today is the skin can actually process what are considered aroma molecules through recently discovered olfactory receptors. So you might be thinking to yourself, wait a second, there's a lot of skin senses. I kind of understand some of these. I didn't know that the skin could actually smell, but I don't want to give too much away. So our two presenters that are going to be with us today, first we have up from our very own global education team out of Los Angeles, California, and that is Tara Vasquez. She is our global curriculum developer for the International Dermal Institute and Dermalogica. And she's responsible for postgraduate curriculum for US and our international markets. And she has over 15 years of industry experience as a therapist in both traditional and medical facility settings as well. And she just also gives great instruction and education for the brand. So we're going to be happy to have Tara with us, who's going to be presenting to you on the first part of our webinar. Then joining us, we're so excited to have an expert from our Unilever location. This is Dr. Jian-Ming Li. And after Dr. Li obtained his PhD, he actually went on to DuPont to research for his postdoc in maize genomics. Now, it doesn't stop there. He actually spent years investigating vitamins and human health before he joined Unilever in 2001. And at Unilever, Dr. Li has led a number of skincare research projects that include diagnostic, age management, and recently sensory biology, which is all about our topic today. So we're going to be excited to have Dr. Li join us here a little bit later on with Tara. So Tara, I'm going to hand it over to you to lead our team today in this amazing topic that you've created with us around skin senses, the power of olfactory receptors.
Thank you, Beth. That was a wonderful introduction. I'm so excited to be here with everybody today from around the world. You know, as Beth said, I'm Tara Vasquez and I'm part of Dermalogica's Global Education Department as a global curriculum developer. And it's so fun to be able to have my hands in new science and be able to share really great information with everybody. So you may be familiar with some of our work out of global education. We launched our award-winning Clean Touch course for both skin therapists and makeup artists as a great way to stay safe during this pandemic. So if you haven't taken that, you might want to check it out. And I am just really, really looking forward to sharing this new research around olfactory receptors in the skin, because it is in its infancy and we are able to learn and kind of hypothesize about how this can greatly impact our skincare industry. So today we will be reviewing our body's senses. So we're going to talk about the traditional 5 senses that we are all very familiar with and more. Plus, we're going to learn how our body's able to perceive those senses with a little bit of background on sensory receptors. Then we're going to move into our area of exploration around new science with olfactory receptors in the skin. And we're also going to discover some really cool examples of how this discovery could possibly shape the future of skincare. And like Beth said, we will have a really good Q&A session with Dr. Jian-Ming Li after we go through all the slides. So I'm so excited to get started. And we are going to start off with what we do know, the 5 senses. So traditionally, you know, we associate each of these senses with a specific area of the body, also known as a sense organ. So sight or vision is the capability of our eyes to focus and detect images of visible light, varying colors, hues, and brightness. So our visual perception is how the brain processes these impulses. Hearing or audition is the ability to perceive sounds through our ears by detecting vibrations. And again, this is how our brain perceives these senses. And smell or olfaction is our ability to detect scent and aroma through our nose in the form of chemical odor molecules in the air. And then we also have taste. So taste is connected to our mouths and our tongue. This is also known as gustation, and it's how we are able to taste substances of our food. But it's also one of the ways that our body protects us, because if we're accidentally having something that's not good for us, like something that could be poisonous, our taste buds are going to tell us that that is bad and we should not ingest it. And then touch. I think as skin therapists, we are all very familiar with our sense of touch, also known as somatosensory. Hopefully I pronounced that correctly, but that is the perception of our activation of our neural receptors in the skin. And that also includes our hair follicles, along with a variety of pressure receptors, which we will be talking about a little bit later. But did you know we actually have 7 senses? So you'll see here on the screen this pink circle with our little stick figure on a ball. So vestibular sense is our sense of balance. So this is how we are able to feel when we're moving forward, where our head is at. It's why you can stand on one leg. You can't see me, but I'm standing on one leg right now. And then our other sense is called proprioception. And this is all about knowing where you are relative in your space. So let's do a fun activity. I can't see you, but I have faith you are all going to follow my direction. I want everybody to close their eyes and hold out their hands. And now touch your nose. So the reason we can do this with our eyes closed is that sense of proprioception. So these senses really play a large role in how we react with our environment and how our brain is perceiving all of the wonderful things around us. So now that we have reviewed our senses, I want us to take a sensory memory test. Don't worry, it's easy. What I want you to do is use the chat box to answer some of my questions. Now, I want to turn everybody's attention to this image of an orange on the screen. And let's start with our sense of smell. When you see this image of an orange, I want you to imagine how it smells. So that means closing your eyes and taking that big imaginary whiff. What do you think that would smell like? And I invite everybody to add some of those descriptive words on the smell of an orange into our chat box. Seeing lots of good answers coming through. So we've got acidic, fresh, sweet, tangy, invigorating. Oh, I love that one. It is very invigorating, the aroma of an orange. Citrusy, awakening, absolutely. Uplifting, yes. These are great words to describe the smell and how it makes what you're perceiving and how it makes you feel. Okay, so now let's think. When you think about what an orange looks like, what are some of those descriptive words that you would want to tell somebody when you're describing what it looks like? Let's see those words coming through our chat. Bright, bright and inviting. We're seeing that it's round and plump. It looks mouthwatering, vibrant, orange. I was waiting for that one to come through. It looks orange. It looks sunny and bumpy. Yeah. And now I want you to think about touching and holding that orange in your hand. How would you describe the feeling of this orange? Heavy. That would be a very juicy orange. Cold. So we like our oranges in the refrigerator. Firm, cool, uneven texture, bumpy, like a softball. Round, bumpy, and squishy. These are really great words. Okay, and the last one we're going to go through is imagine tasting an orange. What words would you use to describe its flavor? Mouth-watering, tangy, juicy, fresh, sweet. These are some great words. For those of us where it's early morning, I think it's time for a glass of orange juice. Fantastic. Thank you guys for participating. This is really fun. So to recap our activity, we talked about smelling the familiar citrusy aroma. In your mind's eye, did you see the vibrant orange color? Do we feel that soft and rippled rind? A lot of people said they were heavy, so nice and full of that orange juice. And then finally that taste, that sweet, tart, juicy flavor that just gets us, you know, our mouth watering because we want to have a bite of this wonderful, refreshing orange. So the point here is that our senses allow us to experience the world around us, but they also help to easily evoke our memories. And even without the physical orange in our hand, we all had a clear memory of the sensorial experience from an orange. And this proves that our senses are extremely powerful. They're very powerful, and it really helps us to navigate through our world. So now you're probably thinking about your senses in a whole new way. Just that simple picture of an orange really can, you know, bring on this activity that helps us to dive deeper into what do our senses do? And you're probably thinking at this point, how do we process all of these senses? This is a lot of information that we just took in thinking about an orange. So basically, we process our senses through sensory receptors. And your next question is most likely, what is a sensory receptor? Well, a receptor is a protein molecule to which a substance can bind, causing a change in the activity. And we love to say, this is like a lock and key, because each receptor has a unique stimuli that can activate it, just like each lock has a unique key that can open it. And I love this image because there's lots and lots and lots of locks on this famous bridge, and each one of them needs a key to open it. And that's like our bodies. We have so many of these wonderful receptors, and each one has that specific stimuli which activates it and then allows our body to perceive our senses. So to take this a step further, I want to talk about 3 types of sensory receptors. The first one here are mechanoreceptors. And these are most commonly associated with the skin, and they help to let our bodies know the levels of touch and pressure, along with our sense of temperature and pain. Now, as skin therapists, we know this one, but we also have photoreceptors in our body. And these are typically associated with vision, so our visual photoreceptors. But I have a great example of a non-visual photoreceptor within our skin coming up. And also we want to just touch on chemoreceptors. So chemoreceptors really involve the detection of chemical stimuli that allow us to taste and smell. So let's get started with those mechanoreceptors. Mechanoreceptors respond to mechanical stimuli. So let's start with the touch and pressure. So our tactile receptors respond to different levels of pressure. And here you can see in this diagram, we have some of these spotlighted. So our Meissner's corpuscles and our Pacinian corpuscle are responsible for pressure and vibration sensations that we feel through the skin. As far as experiencing that deep pressure, that would be the Ruffini's corpuscle, and something like that very gentle touch would be the Merkel's discs. So these minute organs really help to pick up contacts with the outside world and feed that information to our brain. And this is why we could feel a little pebble in the bottom of our shoe, or even maybe like a little, at a barbecue or something, a little fly on our skin, so light. We can feel that through our sense of touch and pressure. And this sense of touch is very spread out over our body because it's spread over the entirety of our skin, which we all know is our largest organ. And in addition to, don't want to forget about this. So you may be familiar with Dermalogica's Neck Fit Contour Serum, where you roll this on your skin and we are actually physically activating some of those mechanoreceptors to help firm and lift our neck. And then we also have our free nerve endings in the skin. And these help us to have the sense of temperature and pain. So specific to temperature is our thermoreceptors, and these are stimulated by heat and cold. And something interesting to know, a fun fact, is that we actually have about 3.5 times more cold receptors than we do warm receptors. And we also have something called nociceptors. And these are the ones that are responsible for telling us when we are in pain, whether it's from heat, acid, too much pressure. Has anybody ever slammed their thumb under a hammer? And also too much cold. So this is really interesting because our thermoreceptors tell us when things are hot or cold, but our nociceptors tell us when that heat is too hot and it becomes painful. And that's actually around 45 degrees Celsius or 113 degrees Fahrenheit. So these actually can kind of work together when it's necessary for our bodies to be aware that something could be harmful. And now we're going to talk about our photoreceptors. And basically, it just means that we have receptors in our bodies that can respond to light stimuli. So here on the screen, we kind of have that blown out section of the visible light. Now, our visual set of photoreceptors are typically talked about as the rod and cone receptors in our eyes, which respond to that visible light spectrum that was on the previous slide. And this we're pretty familiar with, right? So our rods have high sensitivity to light function, and they're really great in night vision And then our cones have a low sensitivity to light, but they're more responsible for that color vision. So together they play a big role in what we perceive through our eyes. But did you know we also have a non-visual photoreceptor, and this is melanin. So melanin is considered that non-visual receptor in the skin, and melanocytes are the skin's photosensory system. So I just found that this was an extremely fascinating fact, because we know that when we're in the sun and our melanocytes kick into gear to produce the melanin to protect us, that's a great example of how we are having that non-visual reaction or perception going around us. And then on to our chemoreceptors. So chemoreceptors respond to chemical stimuli, and this is really about the sense of smell and the sense of taste. So when it comes to our sense of taste, these are also known as our gustatory receptors, and they are typically found in the mouth. Now, we have 5 different tastes that help us to kind of understand what's going into our bodies. So when we taste sweet food, this is to help us recognize energy-dense foods like honey, strawberries, cake, lots of calories, energy-dense, if you will. We can roll with that, right? Cake can be like that. Fruit juices. And then when we smell sour, this is actually, it's the taste of acids. So we're thinking about vinegar and lemon juice, maybe cranberries, yogurt or buttermilk. And then we can also taste salty foods. So this would be like table salt. So we can think of things like soy sauce, processed meats, preserved olives. I love a good olive. French fries. And we also can taste bitter. So bitter would be things like coffee, wine, dark chocolate, arugula lettuce. And then we also have something called umami, or savory. And think about like meat broth, aged cheese, ripe tomatoes, even something that might be a little bit spicy. Umami is also known as just kind of being that overall great flavor. When you eat something that truly embodies all of these things, it's also referred to as umami. And then actually your body tends to feel more satisfied because each of those taste receptors have been activated. And for smell, we have our olfactory receptors found in the nasal cavities. So remember, we think of our nose as a sense organ. So your ability to smell comes from these specialized sensory cells, and they actually connect directly to the brain. So there's actually 10 basic smells that we can detect. So I'm going to give you guys a moment to think about what kind of basic smells or categories, if you will, that we can detect in our noses. And I'll give you a hint. An orange embodies one of these scents. So I see smoky, acrid, sweet, acid. Good. So the human citrus, there you go. Somebody caught on. Unpleasant. Yeah, there are some unpleasant scents. Tropical. Oh, putrid. You guys are on top of it today. So our smell, our sense of smell, we can smell things that are fragrant, like flowers and perfumes. We can smell things that are fruity, like all of our non-citrus fruits, such as maybe an apple. Then we can smell citrus, like lemons, limes, and oranges. woody and resinous. So pine and fresh cut grass, and chemical smells like bleach, ammonia. Ooh, and sweet smells like chocolate, vanilla, and caramel, especially with holidays coming up. We can smell things that are minty and peppermint, like eucalyptus or camphor. Toasted and nutty, so peanut butter, almonds. Pungent, like blue cheese. I love a good stinky cheese. Cigar smoke, and decayed, rotting meat, sour milk. And these are considered the 10 basic smells. But what's really interesting is that our olfactory receptors actually work together. So it's said that we might actually be able to detect up to 3 trillion individual smells, because these come from combinations, and our olfactory receptors are sharing information up to our brain. And I hope this really just kind of opens up your mind to the ability of our olfactory receptors. I find it fascinating, the intricacies of the body and the skin, and learning so much more to prepare for this class has been really interesting. Oh, onions and celery greens. Yes, yes, yes. So a fun fact to follow this is, did you know that flavor is a combination of taste and smell? So if you think back to our activity with the orange, we had a lot of descriptive words that could have kind of covered both taste and smell, which also, relate to the flavor. So when you think about that flavor of something, it has a lot to do with the aroma coming in and the taste. So a nice cup of coffee in the morning, oh, it's the best, right? So now that we have kind of reviewed our senses and our sensory receptors and gotten us excited about the intricacies of how our body gets to perceive the outside world, let's switch gears and take a look at some new science that is available. So we now know that there are olfactory receptors beyond our nose, and they have discovered these receptors in our skin. So this is an exciting new area of study. It's definitely in its infancy, and that's why we are so excited to bring it to the forefront and let all of you know what's going on in our exciting skin industry. So let's start off with the discovery moment. So I do remember reading about this a long time ago in 2014, and I thought, this is so interesting. And then we had a really fun conference, and it was brought up again. And that's really what sparked me to dive into the subject more, was this very first discovery. So scientists discovered an olfactory receptor known as ORT4 in the hair follicles outer root sheath, and that this particular olfactory receptor, or OR for short, is stimulated by a synthetic form of a sandalwood odorant known as Sandalore. And remember that lock and key analogy? So the OR to AT4 is opened and activated and stimulated by the key of the Sandalore. So this is the specific receptor and stimuli connection. And what they found when they applied it is that it helps to promote epidermal keratinocyte migration and proliferation. And when this happens, it actually increased human hair growth by prolonging the anagen phase. So where do you think they went with this? They thought, maybe we could create a treatment for hair loss. Now, as a professional skin therapist, I would like a treatment to get rid of hair, not grow hair, but that's another day. So this is the study that really sparked it all when we learned that there are the OR receptors in the skin and that Sandalore is a specific activator for the OR2AT4. Now let's take a little bit of a closer look at how this works. So we know that this is going to help promote wound healing because it helps with the cell proliferation. Now, hopefully you guys can see this large enough on your screen. But you can see on the left side, kind of that magnified area of those little green olfactory receptors on the root sheath of the hair. And here where they start to activate OR2A4, you can see that this follows through down to the Krebs cycle. And the Krebs cycle is where we create that energy for our cells and we start to increase that ATP production or adenosine triphosphate, which is the energy that all cells live on. It's like currency in our body. We use it and we have to replenish it and we use it again. And then we also found in hair development that when those receptors are activated, it follows through that complicated pathway to induce the anagen phase and prolong the anagen phase. And actually prolongs the anagen phase because it stops the cell apoptosis, which allows the hair to continue to stay there. Fascinating. This is just the tip of the iceberg when we are starting to learn more about our olfactory receptors. So in addition to our OR2AT4, which we know can be responsible for wound healing and hair growth, we also are going to take a look at the olfactory receptor 5,1E2. And this has to do with melanocyte differentiation and pigmentation. Then we have OR2A4 and OR2A7. So it's written as OR2A4/7. And these two receptors actually help with cell proliferation along with the secretion of interleukin-1, part of our anti-inflammatory system. And then we'll also briefly touch on OR51B5, which helps with cell migration, regeneration, and again, IL-6 secretion. So jumping into that OR51E2, what is so interesting about this particular receptor is that it's found on the surface of melanocytes. So that means this can actually help to influence pigmentation disorders. Can you imagine what we can do with this science? And that it also helps to influence melanocyte homeostasis. So here you can see in this red circle that the OR51E2 has a direct link from the melanocyte to our brains. And this is just in the infancy. I keep saying that because there's so much more we are still continuing to learn. And this is fascinating that we may be able to have a better handle on influencing pigment disorders and melanocyte homeostasis in the future. We just really need to find that key, that specific activator to unlock the wonderful benefits. And then we also have the OR2A4/7, and this set of olfactory receptors are stimulated by something called cyclohexyl salicylate. And then we have OR51B5, which is stimulated by isononyl alcohol. So the reason that these two are on one slide is because they have similar function, but a little bit different. So there was actually 100 structurally different odorants that were screened to try to figure out what the actual receptors are what the actual stimuli are for these particular receptors. And we found out with OR2A4 and 7 that this helps to influence cytokinesis, or our cell division. And on top of that, it also helps to regulate the rate of cell proliferation, as well as secreting IL-1. And these two receptors are found in our keratinocytes and in our basal melanocytes as well. So there's the melanocytes popping up again. Whereas our OR51B5 helps with cell migration, cell regeneration, and the secretion of IL-6. So together, these really have a lot of influence on basically our cell turnover rate and having those interleukins in there to help with our immunity. And so what we're really gathering from this science is that olfactory receptors within the skin are able to influence barrier homeostasis, cell proliferation, migration, and regeneration. And a comprehensive understanding of sensory receptors in epidermal keratinocytes could lead to a new paradigm shift of the human healthcare system, as well as how we are treating the skin. So I hope everybody is, as nerdy as I am and thinks that this is just the most amazing science, and we can't wait to see what can happen within the future of skincare. So before I get into some of these examples, I just want to remind everybody that this is kind of the section where we're going to hypothesize and think about what the wonderful possibilities are. But the truth is, we don't have all the evidence for each specific activator or stimuli. So we want you to kind of take note and think about these examples, because moving forward in our industry, we may start to see these pop up. So of course, the first one we're going to talk about is Sandalore, which was involved with that discovery moment. So we know that Sandalore is a synthetic form of the sandalwood aroma molecules, and that it was originally found to induce hair growth. Remember, it helps to prolong that anagen phase. And also something that was interesting with this discovery is that it needed to be continually applied to support this induced hair growth. But because it aids in cell proliferation, we are also, able to connect this to wound healing. So activating OR2AT4, which is the olfactory receptor identified within human skin, helps to signal an increase in calcium and cell proliferation, which can speed up the wound healing process. How amazing is that? How amazing that we have an olfactory receptor in our skin, and that's going to help with wound healing. Now, in the beginning, we loosely say, can the skin smell? So I think by now you guys have caught on that our skin isn't actually smelling these aroma molecules, but it still is able to bind to these aroma molecules. And the way that our mind is perceiving them from our nose is a little different than the way our mind is perceiving them from our skin. Same odorant molecule, different ways our body is using them to function on a very healthy level. And of course, I'm going to talk about lavender, right? Lavender is known for having this calming effect on the mind. We love our lavender in our treatment rooms. We love our lavender at night when it's time to go to sleep. But also lavender is known to be very healing on the skin. It helps with anti-pain and anti-inflammatory influences. So lavender has many different applications for our daily lives. But I just wanted to share some of these share these two common lavender odorant molecules. So we have lineal acetate, and this actually has lavender and bergamot aroma. And, you know, when you guys are looking at ingredient lists, you might see lineal acetate on there on its own. And that's because sometimes it is required by regulatory to have these constituents listed. And I'm sure most of us are familiar with linalool. So linalool is actually found in over 200 plants besides lavender. So keep in mind, just because you see that word linalool on an ingredient list, it's not always tied specifically to the lavender. And here's a great example that I kind of stumbled upon, and I'm very excited to share. So phytonsides. So basically, if you've heard of oud oil, which is a very kind of treasured perfume that is extracted from wood and trees and sap, basically phytonsides are loosely considered essential oils or volatile organic compounds that are released by the trees. So if you love to go on a walk or a hike in a forest, also called forest bathing or shinrin-yoku in Japan, and then after you go on this hike, you just feel replenished and a little happier, less anxious, less stressed. You know, that's the phytonsides. The trees are helping us, and our skin is able to connect with those phytonsides. So I just thought this was an incredible area of study to continue to look into. I don't know about you, but I'm probably going to definitely be going on a hike this weekend, get all my phytonsides in. And so when it comes to skincare, phytonsides are wonderful because they have an antimicrobial effect. They have a strong antioxidant influence, and they're also anti-inflammatory. So phytonsides are wonderful because you know what they do? They really are part of the trees and other plants' defense systems. So the way that they can work with our skin and help to enhance our own skin's antioxidant defense is just so fascinating. And the sky is really the limit. So keep an eye out in case you, in case you start to see these pop up in skincare. And our last example here is rose. So Rosa centifolia, also known as Rose de Mai, the Provence rose or the cabbage rose, because it has really tightly packed petals kind of resembling cabbage leaves. You may see this a lot in skincare. You also might be familiar with the Damascus rose, also known as the Bulgarian rose, the Turkish rose, or the Arab rose. And of course, with our Dermalogica, if you're familiar, we have used rosehip seed oil in some of our products. And typically you'll find that these rose ingredients are in our formulas as an essential oil or a hydrosol or a hydroessential. So when they are creating the essential oil, there is the water portion and then that highly coveted essential oil. But that water portion is also really great and has a lot of benefits. So it doesn't matter whether it's oil or hydrosol, that rose constituent is still going to be really beneficial for our skin. So the way that this might help in skincare is that we know rose has a very calming and relaxing effect when it is inhaled and breathed in. It also has a strong antioxidant effect in the skin. And we also know through transdermal application that it helps to reduce epinephrine-induced stress. So when we're under a lot of stress and our hormones are kind of going crazy and our skin starts to show it, rose actually can help to calm and reduce this. So those are just 4 examples of some well-known and some new types of aroma molecules that can possibly be connected with our skin. So I hope that you guys are getting, it looks like the chat is really blown up and people are into this like me. So fascinating. So at this point, I would like to invite our expert guest, Dr. Jian-Ming Li he is a sensory biologist from Unilever, and he has been an integral part of helping me to have a great understanding and really learning more about olfactory receptors in the skin. So I think from here, we might want to invite him in. Hi. Oh, Dr. Li, you are on mute.
Yeah, can you hear me now?
Yes, we can. Yay.
How are you doing? Good.
I'm good, good. How are you doing today?
Very good. Thank you.
Thank you so much for joining us. I can tell from the chat that our students are loving this in-depth scientific class and they have a few questions, but first, I have a few questions for you.
Sure.
So I just, again, want to thank you so much for sharing research with me and helping me to have a better understanding and really get into all of this great science. So it's time to drill you. Are you ready?
Yeah, sure.
Okay.
Go ahead. Now, before you go ahead, I do want to make a comment.
Okay.
Because you have shared some very excellent examples, some of the essential oil fragrance that have a skin effect, right? For example, lavender, that obviously very nice calming smell, and has been shown to have an effect on skin in terms of anti-pain, anti-inflammation. However, it is not known whether this effect goes through skin olfactory receptor. I just want to make people understand, although some of the fragrance may have a skin effect, it may or may not go through olfactory receptor per se. So that's why we need to understand the mechanism. Just want to make that comment. Okay.
Yes. Yes. So like Dr. Li said, we're still learning if some of these wonderful, skincare ingredients we're familiar with have that direct link to our olfactory receptors. So we just need to keep our eye out for the latest science and see what's going to come up. So my first question for you is, can you share any limitations you found in the studies for olfactory receptors in the skin?
Olfactory receptor is a GPCR, so-called a G protein coupled receptor. So in that sense, it's not any special than other GPCRs. Now, as probably many people know, that GPCR is one of the largest protein that the drug company is very actively looking at for drugs, right? However, there is a differences between skin and then neural in terms of olfactory receptor expression. For example, in our nasal epitheliums, the neuron that actually express the olfactory receptor, one single neuron express one single olfactory receptors, very special, unique. However, in the skin, the one skin cell could express multiple olfactory receptors. So that actually presents some challenges in terms of study the mechanisms. Now, there are other challenges that we have in terms of study olfactory receptor in skin. For example, many skin olfactory receptors, as we know, they have not been deophanized. When we say they have not been deophanized, means we don't know what are the specific agonist or antagonist for this particular olfactory receptors. Because of that, making the mechanism study very, very difficult, because you don't know, you can't say inhibit this particular receptor or activate this receptors, especially in skin when each cell express multiple receptors. right? So the second challenge is that olfactory receptors have many of the olfactory receptors actually very similar to each other in terms of sequence. So it's very difficult to raise high quality antibodies to specifically targeting a particular receptors. Because of that, it's difficult to do like immunohistochemistry type of analysis to see where the receptor expressed in which part of the skin, for example. So also, although we have some knowledge about olfactory receptor in skin for some of them, but many of them are only studied in cell lines or animal model. It's very, very few that we have knowledge in terms of what they're actually doing in skin in vivo. So that obviously is another challenge that we have. You can't just from like what do you smell or what the olfactory receptor does in your nose to kind of trying to deduce what is potentially done in skin. So we have to kind of try various different ways to identify what they could potentially do in skin. And then once we understand what they do, then we're trying to understand the mechanism and how it works. So those are some of those challenges for sure that make it harder to study.
Yeah, I remember reading in some of the studies that they were trying to find people who have anosmia, if that's the right way to say it. So no sense of smell in order to help kind of, like you said, de-orphanize those OR receptors so that there is no olfaction in the nose happening and it's only in the skin. But that's very difficult to do.
Right.
So yes, okay. Thank you for sharing.
No problem.
So then my next question for you is, and I'm sure a lot of our viewers are also wondering this. Can you explain the naming process for each OR? For example, what exactly does OR2A-T4 mean?
So I mentioned earlier, the olfactory receptor is part of so-called the GPCR family. In fact, olfactory receptor family itself is the largest family in the human genome. Now, this superfamily contains about 400 functional genes. Actually, we know their function in the human genome, right? And then about several hundred more pseudogenes. Actually, they do not function in human genome. Now, they have a diverse sequence, but can be assigned to families and subfamilies on the basis of their sequence relationship. For example, if the two olfactory receptor have sequence similarity more than 40%, then they are classified into the same gene family. So there are about 18 olfactory receptor gene families. And then if we found that two olfactory receptors have more than 60% of sequence similarity identity, then we put them into a sub family. There are actually more than 300 subfamilies. So a very complex system. That's why we need to naming them. Now, how do we naming them? What we basically do is say OR means olfactory receptor. So in the case of OR 284, then you have olfactory receptor 2 means the numerical number means the family 2. Subfamily AT and a member number 4. Now, like I mentioned, there are 18 olfactory receptor gene families. So in fact, we have gene family 1 through 14, and then jump to gene family 51, 52, 54, 56, right? So the one example you mentioned, Tara, in your seminar is that there is an olfactory receptor 51E2. So it's not like we have 54 or 51 gene family. In fact, we only have 18 gene family.
That is, I cannot even imagine the family tree that we would have to create to go through all of these olfactory receptors.
Yeah, but one comment I want to make again is that usually because of sequence similarity, right? We know that if the two protein that have similar sequence, then oftentimes that means they may have similar functions. So that's applied the same to here, that if the two olfactory receptor like in the same subfamily, they're often recognized structurally related odorant or fragrance.
This is so fascinating. I think we're going to have another session together and then go even more into depth. Thank you for sharing that.
No problem.
So next I have for you, when it comes to skincare, can the olfactory receptors in the skin tell the difference between a lab-derived ingredient versus a natural stimulus?
If your natural stimulant means the natural ingredient, then yeah, I would say because in principle, the olfactory receptor is a receptor like you mentioned, lock and key. So as long as the key is the same structure, then they're going to open the same receptor, right? So in this case, if the synthetic compound is the same as the natural compound, then they do the same thing. However, we know that natural ingredient, when I say ingredient, oftentimes means a mixture, that they often contain multiple components such as structurally similar or maybe diverse compound in the mixture. In that sense, they may have a different characteristics to kind of bind to the receptor differently. And so in that sense, it may be, but also natural ingredient oftentimes have some small structural modification, like methyls or maybe some sugar, modification to the compound. Therefore, they change their binding affinity to the receptor. So in that sense, natural and lab-derived ingredient may behave differently.
Very interesting. So back to that lock and key metaphor, as you were explaining, I was thinking about when you have another key made, when you have like a duplicate key made, a lot of times it works, but sometimes it doesn't quite work because it's just not cut exactly the same as the original.
Mm-hmm.
The lock and key, always coming back to that metaphor.
Yeah.
Okay.
But one comment I also want to make is just to add some complication here is that although we call this lock and key, but the olfactory receptor, the same olfactory receptor may bind to multiple odorant or fragrance. It's not only bind to one. And one fragrance compound could also activate multiple receptors. And that's actually combinatorial effect is the reason why we can smell billions of smell compounds, because otherwise we only have 400 olfactory receptors. If only one by one, then you can only smell 400 compounds, but in fact, that's not the case. And then because of combinatorial effect, this 400 olfactory receptor can be activated by many different compounds, and then they result kind of smell billions different smells.
So interesting. This is just such a cool area of study. Thank you for sharing that.
Right.
And our next question for you is kind of more of a personal question for you, but what do you find as the most interesting thing about studying sensory biology?
So in terms of the interesting, in terms of sensory biology, I would say for one, I'm very fascinated by what are the endogenous ligands for skin olfactory receptors. And so the reason I say that is because, in fact, evolutionarily, olfactory receptor expression in skin preceded smell, the sense of smell perceptions. Because some of the low organisms do not have the brain to smell and to respond, but they already have the olfactory receptor expressing skin. Now they could be doing two things, right? One is the skin itself as a sensor organ to detect food danger in the environment. But because they some of the signals are actually quite low in concentration, and especially when you have this kind of function in terms of handling hair growth, they may actually have endogenous ligands to do that when the skin was damaged or exposed to the external environment. So what are the endogenous ligands that are activating those receptors and then result some of the functional response? It's quite fascinating. And I want to use one example like OR 284 as the example you mentioned. The 2R OR 284 actually can be activated by Sandalore, for example, and then in the skin keratinocyte cell. But once the keratinocyte cell being activated, it actually releases ATP as a neuron transmitter, and then activating a neighboring neuron cell. That this interaction between the keratinocyte cell and the neuron cell, that is how actually increase or enhance the wound healing process. Yeah, so that's kind of local effect. But then back in the question, when you have this sensing neuron cell being activated, does it actually transmit signal to the central nervous system, the brain, to induce some kind of body response? So that's a lot of questions behind it that we don't know the answer yet.
Wow, yeah, you're right. That is a lot of thought, a lot of interesting aspects to continue to explore.
Yeah.
And then also, what area of sensory biology in the skin are you working on next?
Yeah, no, this is exactly what I said. Some of the very interesting aspects that to me, not just about the olfactory receptor does in skin itself, but I'm also interested in whether olfactory receptor actually being in the skin as a sensor to detect environment, and then that transduce the signal to the body, other part of body, and to the brain. So the skin, body, mind connection, and therefore, what are the sensory neuron play in this connection? It's very fascinating. Something that I'm working on and continue working on now. So we know that the skin being the interface between body and environment, it plays a critical role in terms of detecting signal and transmitting it to the body and brain. And then I believe that not just about olfactory receptor, but other type of receptors, such as touch receptors, the ping receptors. Those some of those we already know that when we touch, we feel it in your brain. The ping obviously doing the same thing. So the question is, does the olfactory receptor does the same thing as well? What exactly signal is transmitting?
Very interesting. And then one last question for you. Can you give us any examples of how this is already happening? What are some examples of where we can see these studies right now in our industry?
Yeah, so again, in terms of the skin olfactory receptors, the field is still very early infancy. But the best example is OR 284. You already showed that in your seminar. So we know that activation of OR 284 stimulate hair growth. In fact, inspired by this work, there's a company called Actera that already have a market product in the field, in the market for anti-hair shedding and hair regrowth. So that's kind of the olfactory receptor field. Obviously, if we go, outside of olfactory receptor field, but still within this kind of sensory biology and chemoreceptor field, and the best example is the pain receptor TRPV1. And this mechanism has been very well studied. In fact, this year, if people, hear the news, is that the Nobel Prize in Physiology Medicine awarded to the science of the stomal and pressure sensation, including the TRPV1. So many pharmaceutical company actually is very active working on this particular receptor to develop drugs for various pain, dealing with pain issues. So this definitely is a very, very exciting field because it's in the early phase especially for olfactory receptors skin. There's a lot of potential opportunity as well.
Fantastic. Thank you so much for sharing that level of, science with us and really helping us to hear it, straight from you as our expert. So at this point, I just want to say thank you again for not only helping me, but also joining us today and answering some of our questions. And I think at this point, I'm just going to invite Beth to come back on. She's going to share any other interesting questions from our chat.
Sure.
So welcome back, Beth.
Great, thank you. Thank you so much. Wow, like what an amazing presentation, what an amazing topic. I think everyone is just kind of blown away about the science that's out there right now, and also very excited, to say the least, to definitely learn more on this topic. And thank you, Dr. Li, for joining us today with Tara to really dive into some of those meatier questions that we knew we would have. And Tara, fantastic job about bringing this to life with everyone. So we do just have a few questions. I think if we could take a few moments, Dr. Li, of your time, I think that will be exceptional. One of the questions was how, on olfactory receptors, how do you actually, I think the word would be find or identify an olfactory receptor? And do you think that there could be more in the skin that have not yet been found?
Yes. So the faster way is that we go into some skin cells, for example, skin keratinocyte cell, skin melanocyte cell, or skin fibroblast cell, and then we extract their RNA from that cell, and then we run so-called RT-PCR, gene expression analysis. And based on that analysis, we can determine whether the skin cell express receptors. Now we can obviously depending on how much you want to do, we can run the whole, 400 olfactory receptor in, say, for keratinocyte cells. So we can determine how many cells olfactory receptor expressed in keratinocyte cell, right? By doing that, we can do that, we'll get this kind of knowledge. But then the question is whether they express in skin, because this is a little bit different, because when you culture the cell in petri dish, they may express different type of olfactory receptors. So now with that information, we go to the skin. Now, when you go to the skin, the challenge is becoming is that you kind of, if you take like a punch biopsy or sometimes a skin biopsy, it's a mixture of cells. You don't know where they express in keratinocyte cell, maybe in fibroblast cell, melanocyte cell, right? And then you have to kind of maybe want to know which part of the skin express of those receptors. Then if you have antibody, that would be really, really good, because then it'll tell you where expressed in the skin. But like I mentioned earlier, one of the challenges that oftentimes we don't have a very high quality antibody do that. So that's one of the challenges. But there's a way of determining the olfactory receptor.
Okay, fantastic. And I think it's definitely not a one direct path to be able to locate those. Do you think that, especially with the fact that you're, how it's being expressed and how you're testing, that there's probably, do you think you could say that there could probably be, more out there that haven't been discovered yet?
Oh yeah, of course. So that's expectation, right? Because like I said, when you look at the keratinocyte cell in vitro, it may express certain number of olfactory receptor, but skin, depending on different conditions, it may actually. So, for example, we know that some of the olfactory receptors involving in the skin and then microbe interaction in atopic dermatitis.
Okay.
So this olfactory receptor may be in one of the olfactory receptor may be induced in this skin condition, may not be, readily detected in normal health system. So yeah, so depending on skin condition, depending on, kind of what interest you have, you probably will see many more olfactory receptors, but it's probably going to be still in this 400 olfactory receptors that we know.
Okay. And that kind of like in all the families and the subfamilies and everything that's going around that as well. Okay.
Right.
So speaking when you mentioned atopic dermatitis, I thought this was an interesting question. We think about reactions, definitely allergies as well. Tara touched on quite a bit about the different types of receptors where our body recognizes, heat or pain. So and linking that into olfactory receptors, could you be allergic to something that maybe you react more when you smell it versus it actually touching your skin? Do you think that's something that's happening out there, or that it could be vice versa, where someone could, if you think about like when people do inhalations, such as peanut allergies, some people have such a high allergy to peanuts, when they smell it or inhale it, it could trigger the reaction, but maybe they could actually touch the peanut and nothing happens to the skin. Do you think that there's some correlation like that with olfactory receptors?
So I'm not so sure. I don't know if I can answer this question completely to satisfactory, but I would say that in skin, because the same kind of odorant or fragrance, that when you touch the skin, it will activating some of the immune response cells that causing some irritation or allergic reaction. That may not necessarily go through the olfactory receptor because of it, or may not because of the olfactory receptor, right? So in that sense, they actually are probably not related to olfactory receptor. So
got it.
I would say if you are allergic by smell, the likelihood that you also allergic to skin is possible, but I would say may not always be the case.
Yeah, definitely could be just individual responses depending on that, and they probably have to dive like a little bit deeper into the trigger or the allergy.
But you have to, exactly. You have to remember that although the skin and the nose, they both express the same olfactory receptor, same structure, but in order for the receptor send a signal, it activates, and then there's a lot of other protein complementary to this receptor need to work together to transduce a signal. Therefore, because those helper protein in the nasal neuron are different from in the skin, that's why they may not transduce the same type of signal.
Okay, so, definitely.
Right.
Yeah. No, I think I know for saying that you're not sure if you can answer it fully, I feel like you did an excellent job. And it gives, I think, everyone's a lot to think about. I know we have, a lot of questions just now, because I think you start opening up to olfactory receptors and then looking at all the other types of senses and how everything is connected. And of course, in correlation to the skin. There was a question too, is that could there often be a chance when an olfactory receptor is overly stimulated? Is there a response to that?
What exactly, can you rephrase this?
So I'm thinking, so I know we talked about there's actually the key or the lock and key that kind of turns on the receptor. When that happens, has there ever been any type of studies that have shown like overstimulation or any type of maybe adverse, reactions to that olfactory receptor being kind of turned on, so to speak?
Oh yeah. So olfactory receptor, just like many other receptors, that sometimes when you have a ligand that has overwhelmed the receptor, it actually called desensitization, desensitizing it. So sometimes when you smell overly exposed to certain smell, you kind of lost detection. But also there's so-called internalization of the receptor. When the receptor exposed to the same stimuli over and over again, actually the receptor expression becoming lower. So in that sense, and then you cannot smell that very well. So in that sense, I'm saying the other way around, if you overly activating a particular receptor, it could certainly potentially have some negative effect. And I can't give you an example right now, but I would say that in principle, it could have a side effect in principle, yes.
Okay, all right, great, thank you.
No
Well, I know we are wrapping down to the end of our webinar today. We've had some great participation from everyone, some fantastic questions as well. Dr. Li, again, thank you so much for being here today. And, just really, again, taking the science to yet a new level. And a huge thank you to Tara for bringing this to life for the International Dermal Institute and our industry members who are so excited to learn more about this topic and see how it moves forward. So we just want to thank you so much for both being here. So Tara, anything else from you? Any last words that you want to share with our group?
I just wanted to say thank you again to Dr. Jian-Ming Li for being my partner in crime to bring this to the forefront for our skin therapists. And I also just wanted to extend a thank you so much. And I also wanted to extend a big thank you to my global education team for everybody's support and assistance while I was working on this passion project. And it really takes, a village over here to bring everything forward. So I'm just so thankful for all of my support and help, and I look forward to seeing where this goes even more in the future.
Fantastic. Great. Well, thank you both. And thank you everyone for being with us today on our industry expert webinar. Again, these webinars are here to just bring you the kind of top level, just kind of scratching the surface on some of these key pieces of information that are happening within our industry around skin science and technology. And we can't wait to see all of you back on our next topic. And again, thank you to Tara Vasquez and Dr. Li for joining us today, and have a fantastic week. And we'll see you next time here on our industry expert webinar. Thank you.
Thank you, everyone.
Bye, everybody.
Bye.
Thank you.